Welcome to this explainer. Today, we're jumping right into a topic that is absolutely reshaping the field of neuro-ophthalmology, evidence-based nutrition in glaucoma. For decades, we've kind of treated glaucoma largely as a plumbing issue of the eye, right? Just lower the pressure. But an absolute mountain of clinical data is revealing a highly complex metabolic side to this disease. So today, we're going to build a clear taxonomy of adjunctive nutritional therapies, carefully separating the rock-solid clinical evidence from the stuff that's purely theoretical.
Okay, let's dive into this. We have 6 distinct areas to cover, and we're going to use a pyramid of evidence structure to process this massive amount of data. We'll start with 1, the new glaucoma paradigm, move into 2, systemic diets and lifestyle, climb up to 3, high-evidence supplements, look at 4, emerging botanicals, examine 5, beverages and chronobiology, and finally wrap up with 6, clinical guidelines and safety.
Section 1, the new glaucoma paradigm. Now, to really understand why nutrition matters here, we have to look at normal tension glaucoma, or NTG. By definition, this is a progressive optic neuropathy occurring with strictly normal intraocular pressure. And honestly, it's a total game changer for how we view the disease. Epidemiological surveys consistently show that 30 to 40% of primary open-angle glaucoma patients actually have completely normal eye pressure. This tells us that glaucoma isn't just a mechanical issue. It's a metabolic and neurodegenerative condition. It shares pathological pathways with other central nervous system disorders. The retinal ganglion cells, those highly demanding unmyelinated axons of the optic nerve, are exceptionally vulnerable to oxidative stress, mitochondrial failure, and localized ischemia. And that, right there, is exactly where nutrition intervenes. Moving on to Section 2, Systemic Diets and Lifestyle, the foundational bedrock of neuroprotection. Before we even think about targeted pills, let's look at the broad, macro-level impact of our daily food choices. And this brilliantly illustrates the point. Let's compare 3 dietary approaches. First, the MIND diet, which is heavily focused on leafy greens and berries. The Rotterdam study found a profound risk reduction here. For every 10% increase in adherence to this diet, there's a 20% reduction in the risk of incident open-angle glaucoma. Incredible, right? Then we have the Mediterranean diet. This provides a robust antioxidant matrix, shielding the optic nerve against oxidative damage, and it was shown to be highly protective in the Sun Project cohort. But perhaps the most striking data comes from dietary nitrates found in green leafy vegetables. These nitrates actually convert to nitric oxide in the body, actively relaxing the trabecular meshwork and improving microcirculation. To put that into perspective, high consumers see a staggering 44% reduction in paracentral visual field loss, which is the specific type of vision loss linked to vascular dysfunction. When we drill down into specific macro elements, the synergy is just fascinating. It's like a highly coordinated defense system. Let's map this out. Vitamins A, C, and E operate together as the ACE index. Clinical data shows individuals with high combined intake are 47% less likely to develop glaucoma, mostly because vitamin C actually regenerates oxidized vitamin E. Next up, calcium and potassium act as a protective shield. High intake is linked to a significantly reduced risk of glaucoma, particularly in normotensive patients. Then we have magnesium. Now, there's a bit of a systemic paradox here. It functions as a physiological calcium channel blocker, offering real benefits in normal tension glaucoma by improving ocular blood flow, but overly high systemic intake might actually lower blood pressure too much, reducing ocular perfusion. So balance is key. Finally, omega-3 PUFAs directly incorporate into ocular tissues, yielding a measurable roughly 8% reduction in intraocular pressure by improving ocular outflow. All right, Section 3, high-evidence neuroprotective supplements. We are now moving up the pyramid of evidence, shifting away from broad lifestyle habits to some highly validated targeted interventions. So the crucial point is this massive dosage right here, 3.0 grams per day of nicotinamide, which is the amide form of vitamin B3. You might be wondering, why such a high dose? Well, glaucoma triggers severe metabolic stress, and that rapidly depletes NAD+, which is an absolute necessity for cellular energy in the retina. Nicotinamide acts as a direct NAD+ precursor, effectively becoming a profound mitochondrial rescuer for sick, struggling retinal ganglion cells. And the clinical data for nicotinamide is arguably some of the most exciting in the entire field. It fundamentally restores retinal ganglion cell bioenergetics. In human trials, we objectively see it improving inner retinal function on an ERG, completely independent of whatever the eye pressure is doing.
Furthermore, phase 2 randomized controlled trials have demonstrated a nearly four-fold higher number of improving visual field loci when nicotinamide is used. But let's pump the brakes for a second. We absolutely must be cautious. This 3.0 gram dosage far exceeds normal dietary limits, and chronic exposure carries a very real documented risk of hepatotoxicity or liver damage. Next on our high evidence tier is citicoline, also known as CDP choline. Typical therapeutic oral dosages for this range from 500 to 1,000 milligrams per day. See, when glaucomatous damage degrades the vital lipid membranes of the cells, citicoline steps in to provide the biochemical substrates that rapidly accelerate membrane phospholipid repair. In multiple long-term clinical trials, it has reliably stabilized visual fields and structural OCT measures. What's even cooler is that citicoline shows incredibly deep biological synergy. When you combine it with other neuroprotective compounds like CoQ10 or vitamin B3, the restorative effects on mitochondrial activity are vastly multiplied. Section 4, Emerging Botanicals and Antioxidants. Now, as we transition to surveying this vast landscape of specific botanicals, it is vital to note that they largely possess a lower clinical evidence grade compared to the high-evidence supplements we just discussed. Let's rapid-fire through these phytochemicals. First, Ginkgo biloba extract. This has shown unique efficacy in normal tension glaucoma visual fields through potent central and peripheral vasodilation, which corrects localized tissue ischemia. Second is Mirtogenol. This is a precise combination of pycnogenol and bilberry. It directly lowers IOP and demonstrates profound pharmacological synergy when acting alongside standard topical prostaglandin analogs. Finally, we have CoQ10 plus vitamin E. Because oral CoQ10 actually struggles to cross the blood-retinal barrier, topical delivery via eye drops is used here to directly support mitochondrial bioenergetics and prevent cellular apoptosis right in the eye. Continuing our survey, let's look at the antioxidant targets. We start with saffron, specifically its active component crossin, which exerts absolute suppression of microglial activation, essentially halting neuroinflammation in its tracks. Next, curcumin and resveratrol. These show immense potential in lab models for retinal ganglion cell rescue and SIRT1 activation, though to be fair, their poor oral bioavailability remains a real clinical hurdle right now. Then we have lutein and zeaxanthin, which are absolutely vital for fortifying the macular pigment, absorbing phototoxic blue light, and neutralizing reactive oxygen species. Lastly, PEA, or palmitoyl ethanolamide. This is an endogenous lipid mediator that offers a dual action benefit through PPR alpha activation. It actively halts neuroinflammation while simultaneously lowering IOP. Section 5. Beverages and chronobiology. Let's move to and see how this builds into everyday patient choices. The daily liquids patients consume actively dictate microscopic intraocular dynamics. Looking first at caffeine, modern genomic research shows a severe danger here. In individuals with a high genetic risk, heavy intake of over 480 milligrams per day results in a staggering 3.9 times higher prevalence of glaucoma. That's huge. Caffeine acts as an adenosine receptor antagonist, which transiently spikes aqueous production. But contrast that with tea. Despite containing some caffeine, the highly protective flavonoids in ECG-C and T result in a lower baseline IOP. In fact, observational data correlates daily hot tea consumption with a massive 74% decreased odds of a glaucoma diagnosis. Now let's compare alcohol and melatonin. The observational data on alcohol is decidedly mixed. Light intake, like a glass of red wine, might transiently lower IOP via osmotic dehydration, but heavy use has zero proven benefit, so strict moderation is strongly advised. Melatonin, on the other hand, is incredibly compelling. Glaucoma patients frequently suffer from disrupted sleep due to the progressive loss of photosensitive retinal cells. Taking exogenous melatonin not only improves circadian entrainment, but its receptors in the ciliary body actively modulate aqueous humor dynamics. This yields modest but very reliable IOP reductions, particularly during those dangerous nocturnal pressure spikes. Section 6, Clinical Guidelines and Safety. Okay, we need to talk about safety, because it is absolutely essential to remember that natural does not mean inert. These supplements carry very real, potent pharmacological consequences. Now, what's really interesting about this slide is just how severe these pharmacokinetic dangers can be if they're left unmonitored. For example, Ginkgo biloba aggressively inhibits platelet aggregation. When you combine that with blood thinners like warfarin or even common over-the-counter NSAIDs, there is a highly elevated risk of spontaneous hemorrhage. And as we noted earlier, high-dose nicotinamide requires strict medical supervision due to the threat of acute liver toxicity. Then there's resveratrol, which acts as a potent CYP3A4 enzyme inhibitor. That means it can dangerously alter the metabolism of other vital medications a patient might be taking, and it also elevates bleeding risks. Because of these profound biological effects, major international bodies maintain a strictly conservative academic stance. As the American Academy of Ophthalmology definitively states, alternative complementary therapies cannot replace medically proven IOP-lowering interventions. Whether we're talking about the Mediterranean diet or targeted citicoline, these are adjunctive, supplementary tools. They fortify the optic nerve, yes, but they never, under any circumstances, replace standard medical therapy, topical hypotensive drops, lasers, or surgical treatments.
We've covered an immense amount of ground today, right from the systemic vasodilating power of leafy greens all the way to the highly targeted mitochondrial rescue of nicotinamide. So I want to leave you with this final provocative thought. As evidence mounts linking glaucoma to systemic neurodegeneration, will metabolic rescue one day become as standard in clinical practice as lowering eye pressure? Thank you to all you focused learners for joining me on this comprehensive academic journey into the metabolic future of glaucoma management. Until next time, keep questioning and keep learning.